Hi, everybody. You can hear me? It's fine? Okay. So you all know that cortical folding begins in utero. And basically, the complex pattern that you can observe in adults are sculpted during the last trimester. And the point is that it is a key time window for the development of cortex architecture. So it's the reason why, for a long time, we assume that we could get out of cortical folding patterns some signature of abnormal development. And we've been developing tools for quantification of the variability for maybe 30 years now. And now I would like to give you some insight about some of these tools via a few applications. And you will see, and maybe it will trigger some collaboration. So it should be noted that the cortical folding pattern is not necessarily completely finished after birth. In fact, at birth, the complexity of the folding is similar as adult folding, but proceed further in terms of getting deeper. And until basically adolescence, where there's a kind of turn of the tide, the folding process go backward because of the myelin development. And this is proceeding further, and the backward process accelerates during aging and sometimes during neurodegeneration. So the yellow picture, there's something wrong in this picture. It's that the yellow picture here, it's not the normal cortical folding process because it's preterm babies, it's not fetuses. And to begin with my talk, to illustrate the first tools, I will try to tackle the idea of whether the folding of preterms is similar as the folding of fetuses. And if you look carefully at this picture, so here fetuses and at the bottom preterms, you rapidly realize that there's a lot of differences. In each column, it's the same age. For instance, you see that the fetus brain is surrounded by a lot of CSF, which is not the case of preterm, which is very surprising. Basically, it means that birth is a very special event for the environment of the brain and probably for the folding dynamics. Another striking fact is that preterm brains are basically globular, while the fetuses ones are kind of flat. The parietal lobe, for instance, is very special. And also, you realize rapidly that the folding looks less intense in the fetuses compared to the similar age of preterm. So, using quantification, we can go a bit further. So we heard already this afternoon about gyrification index, basically the ratio between the surface of cortex embedded in the brain, buried into the fold versus the surface area of a flat brain. And if you look at this chart here, the blue one is for preterms, the red one for fetuses, and you clearly observe that preterms fold faster than fetuses. It's really striking. And you can go further than that into the exploiting this index. For instance, if you compare preterms that are healthy with preterms that had the problem of intrauterine growth restriction, probably at the origin of the birth, here you have two babies with the same surface area, and you see that the intrauterine growth restriction babies are more folded. It's not completely intuitive, because it's not polymicrogyria. It's just that something is speeding up the folding process in this situation. So leading to some interesting biomarkers. And thinking about biomarkers for this kind of features, it's interesting to go to what we've seen already this afternoon, kind of normative charts. Here it's not really normative charts, but it's playing the same role. So the gray triangles are different babies. At the beginning, if it's preterms, not fetuses. So it's just because we keep fetuses away because they live in utero and the folding dynamics is different there. After that, you have term babies and later situations. And here we compare, we put on this chart the black triangles, which are preterm babies imaged at the term equivalent age. And you realize that they are standard in terms of volume and surface area, but their index of verification is a bit lower. Which is a bit surprising because I was explaining before that the preterms fall faster than the fetuses. It means really birth is a key event for folding process. you may be willing to get a bit further in terms of analyzing the difference, for instance, between preterms and standard babies. So there are some tools for that. When you think about the folding of the cortex, it's kind of waves, and when you have waves, you can play the frequency game. So you can decompose this wave into different frequencies, low one, fast one, low, okay, different frequencies, sorry. And I will not go into the detail of this technique developed by David Germano and Julien Lefebvre. You can basically split the world of the frequency into different bands and compute the power of each band, which is its contribution to the whole folding. And you have a band with low frequency leading to what looks like primary folding, basically. And you have further away details coming from the two other bands. And if you apply that to preterms, you can observe, in fact, that the first band, the low frequency band, gets some power very early, followed by the second one, and after that, the third one. It's really looking like the three waves of folding that you heard about in literatures, you know, the primary, secondary, and tertiary folding, even if, of course, it's not exactly that. And it's an interesting tool to get more insight about the difference between preterms and normal babies. And if we compare, we do exactly the same, this kind of normative chart, but with the three band powers, we realize that it's only in the low frequency band power that there's a difference in preterms. They are similar for the two other ones. And basically, this is the first wave of folding, the primary waves, which is occurring exactly at the same time as the birth of these early preterms. So it's making some sense in a way. So basically, we have these tools that allow to assess globally, in fact, the situation of folding. I'd like to focus on something very important when you study folding, which is allometry rules. David is a worldwide expert of allometry rules, and if you're interested, he can discuss with you hours about that. Here, this chart is just showing you the evolution of surface area versus the volume of the brain in humans. And the point is that the slope here is not 2 divided by 3, which would be the slope if it was just a zone, you know, if big brains were looking like the same as small brains. In fact, big brains are more folded. And if we look at real brains, what does it mean more folded? It means that if you look at this frontal lobe here in these big brains compared to this small brain here, there's much more complexity, much more branches, secondary or tertiary fold appearing at the top of gyri. So basically, if you want to compare two groups, for instance, patients versus controls or men versus women, if you do not correct for that, you may be completely wrong in the interpretation of your result. And just one example with extreme case, microcephaly, so brains very, very small, 5, 6 standard deviation from the normal. And here, 3 kinds of microcephaly. The ones stemming from alcohol that we spoke about this morning, and 2 mutations. And if you take into account allometry to assess whether the surface area of these different patients is normal, it is normal. But if you go further into the analysis using index of gyrification, or all this stuff I was mentioning with spectral analysis, you realize that the 3 pathology here do not behave the same. Basically, the fetal alcohol, the FAS, let's say, just follow the even, in fact, trajectory in terms of allometry, while the 2 mutations are either too folded or not enough folded. And it's the only way, in fact, to discover that seriously is to take into account these allometry rules. Okay, so why do we believe that there's a strong link between architecture and folding? Recently, in fact, during the last decades, physicists have shown that with a very simple equation that is modeling the fact that the outside part of the cortex is developing further than the inside part, the white matter. If you simulate that, you just get some folding very similar to the folding in the real brain. I will trigger it again if I can. Okay. Sorry. And if you begin this process with a fetus shape, which comes from Marseille, you get this kind of simulation. And you realize that the morphology is realistic, but the folds are not at the right place. In fact, the reason why folds appear at specific place for each mammal species is probably linked to the fact that there's a lot of heterogeneity in the development and in these growth growth rate. And there are recent results from the last decades from biology showing that, for instance, the difference between a folded mammal and a non-folded mammal is that during the neurogenesis, in fact, there are heterogeneity in the expression of genes leading to the multiplication of cells, of progenitors of neurons. And you have some patchwork of places with high generation of neurons and other ones with less generation, which are kind of protomap of the gyrus and sulci. And basically, this is what is deciding for where the folds occur, even if this mechanics is still behind. So it means that if you have a mutation, for instance, or something disturbing the neurogenesis, it will disturb the folding. I don't know if some of you are specialists of epilepsy. It's very classic in epilepsy to discover some abnormal folding pattern close to the epileptic focus. So now I will speak about another kind of tools allowing to study the local variability of the folding process. To be fair with the RGPD, I'm using my own brain to show you what these tools are doing. So here, basically, it's what you can extract from MRI. It's important to notice, that there's a kind of wavelengths. All the gyri have the same width, which help to understand why a big brain must have more gyri, just because there's some kind of specific rules of the organization of the gyrus in the brain that is leading to this fixed size and this fixed width of the gyrus. So with the computer, you can remove, peel the gray matter and get the white matter, and discover that there are more folding features that are buried in the folds that are important to take into account when you study the shape of a cortical surface. The computer can compute a kind of negative cast of the white matter, which is this blue object. It's surfaces following the center of the fold, a bit like the surfaces you find in walnut if you eat walnut. And finally, you can imagine AI kind of technique, you can recognize automatically the main sulci that are described in the literature. All these colors correspond to a specific entity. And you can finally quantify the shape of this sulci. For instance, you can compute the length of a sulcus or its depth or its surface area. There's another interesting feature, which is the width between the two walls of the fold, which is basically giving you an idea of the amount of CSF in the fold, which is the quantification of the local atrophy, which is mainly used, in fact, to study aging and neurodegeneration. Sorry, I have some window to remove. Okay. And for instance, this has been used to study specificities of the link between, in fact, the local folding amount and various cognition features. For instance, here with preterms from Utrecht, image at birth and at the term equivalent age, all can various associations we have discovered between the size of local fold and some scales of cognition. I mean, there's plenty of questions you can explore with this kind of tool. David has triggered a lot of collaboration to exploit this kind of opportunities. For instance, there's one with Necker with in the context of craniostenosis, leading to process very impressive brains like this one with the huge ventricle. It's just to show that these tools are robust to this kind of situation. There's another collaboration with teams from Nice and Dijon on another mutation changing the cortical folding. If you compare this control here with these patients, you can discover that in the parietal lobe, rather than a huge intraparietal fissure, there's a doubling of this fissure, which is really unusual. You can also study not only macrocephaly, but very big brains. These ones are beyond 7 standard deviation from the normal. And you can see that, in fact, basically the general organization of the classical sulci are still there, even if they are more complex. So there's plenty of opportunity to study the link between folding and all these things impacting cortical development. To train the AI system to recognize in a robust way all these sulci, we had to annotate a lot of brains. And I can tell you that it's much more difficult than any of you can imagine, because in fact, there's not a real understanding of the variability across brain. If you compare these 3 brains, you will realize that they are really different, and we are not sure. We are not always sure of what we label, even if we have this impressive visualization tool. It's really tricky, and I would like to give you more insight about why it's tricky. I would like to let you imagine what it means when you have two different brands without any help to compare them and find a kind of roadmap to go from one of them to the other one. You can easily find primary areas on some of the central sulcus. They are really stable across brain, even if sometimes they can be split in two. If you look at another sulcus, the superior temporal sulcus with its two branches, it's getting much more complicated. For instance, you see that this one is split in the front. There's a gyrus, transverse gyrus here, and maybe it's corresponding to this stuff here, a gyrus that is buried in the depth of the sulcus. This one is interrupted in the middle, and it's a bit the same here. There's a kind of gyrus corresponding here that maybe is the same place. Here you have another gyrus that is outside here and buried between the main part, the main stem, and the green branch. There's a lot of combinatorial variability here, which is leading to a nightmare in the frontal lobe when you want to do annotation. What could it mean? Is it interesting to get some insight about cortical development? It's basically what is driving our research for the last decade. And our idea is that there's in the folding dynamics, different attractors, and depending on brain, the chronology of the maturation of different areas, all kind of things like that, you can go toward one attractor or another one, which means interruption or not interruption of different entities. And of course, you have a combinatorial explosion of these things. So what we are trying to do, maybe I did not say that up to now, each of us has a specific cortical folding pattern, as specific as your fingerprint. And the idea is that we try to decipher that as if there was a kind of hidden language behind. But what is this language? Is it that it's everywhere with everybody the same letter, but different strikes? It's completely unclear, you know. What we have in mind there is it's a bit the differences between all of you when you write letters. We all write a different way. Does it mean something or not in the brains? It's this kind of thing. And when you target this kind of objective, you have to do something which is very difficult. It's distinguish what is normal from what is abnormal. So we are trying to do a dictionary of all the variability of this pattern in the normal population that can be used to assess whether a specific pattern is more frequent in specific pathologies. We first use standard machine learning for that, and I will give you an illustration with the central sulcus. Here you have 30 negative casts of central sulcus, so it's a rather stable shape, but they all have different deformation, different knobs to talk like anatomists. And with machine learning, you can ask the computer to rearrange them so that the ones that are similar are close to one another on this axis. And you realize that now you get on the left the ones who have just a deformation in the middle, and on the right some who are with several deformations. And you can do that with 30 brains or 1,000 brains. You always get the same variability, which is this knob here moving up, letting some space for another knob here. And to understand this organization better, we call that an isomap. You can compute moving average at different places along the axis, and you get an average sulcus. And you see that on the left, you get central sulcus with one knob, and on the right, central sulci with two knobs. And if you do fMRI analysis for population with the different patterns, you realize that with standard motor activations, the hand area is moving up with the knob that I was describing before. And this language area, which is a reading area, is in fact located either close to the sulcus for the one knob pattern, or far away from the sulcus for when it's the buses of the CEA. Far away from the sulcus for the second pattern, which means that this pattern story is really a proxy of differences in terms of architecture. And what happens in terms of abnormality? Here it's exactly the same study with a population with controls and people missing one hand, either because they get amputation or because they have a mutation leading to the development of only one hand. And you don't get the same variability. You get a variability from flat central sulci to standard central sulci with knobs. And what happens is that people with amputations have the same variability as the controls, but people with a missing hand at birth, when you take only the contralateral sulcus in charge of this hand, they mostly have flat sulcus, which is just showing you that this pattern is giving you proxy of abnormal development. And you can even go further than that and do the same kind of analysis with preterms. Here it's with the lateral fissure, using preterms with image at 30 weeks and at 40 weeks. The main variability is a standard asymmetry in the brain between the left and right hemisphere. The back of the lateral fissure is tilted more or less towards the front or towards the back from one hemisphere to the other one. You see the same asymmetry at 40 weeks, and you see it also at 30 weeks. But what is interesting is that if you take the subject, it's the same, so you can correlate both. And in fact, the subjects which are on the left at 30 weeks are still on the left at 40 weeks. It means that this asymmetry is already there at 30 weeks. It's stable. So you can just try to understand the chronology of the way this pattern appear in the brain. And I will finish with what we are doing now. It's basically forgetting about the past, dealing with these things with the most modern AI kind of technique, which are not learning to reproduce what has been done by humans, but which are just reorganizing the data. And I'd like to do an analogy between what has been done with AlphaFold, playing with folding of proteins, with about 200,000 proteins with non-folding, with very, very difficult research to get that. They could now predict the folding of most of the other proteins. In the world of neuroimaging, we have a few 100,000 brains with a lot of other information, fMRI, diffusion, annotation, behavior. And what we are trying to do now is to ask AI to provide us with a dictionary that can be a predictor of all these annotations. Not whatever feature of the folding pattern, but the ones that are really useful to predict behavior or to predict diseases. And I will conclude with two examples of results. The first example is with something that is called autoencoder in the world of deep learning. Basically, it's network that you push to compress like crazy images you give them as input and to ask them to reproduce the images as an output, but they just have a few neurons at the level of a bottleneck, so they can just memorize very standard patterns. They cannot memorize something that is rare. And after that, you can use it to see whether they do some mistakes or not when you provide them with a new brain. And here it's an example in the cingulate area. Here you see a standard cingulate area. The model has been trained with adults, and we test it on children coming from another machine. Here you see a control, a children with a standard cingulate. And here it's a children with agenesis of corpus callosum, which is known to completely modify the folding pattern here. And here it's the reconstruction error for the autoencoder. And you can see that if you take standard adult brains with the same acquisition protocol, you have distribution of errors. The controls, even if they are children, they are in this distribution. But the children with agenesis, in fact, the error is massive, which is just a way to detect that there's something abnormal there. And to finish, another kind of application of deep learning, a bit in the same spirit as with the isomap story before, you can ask the deep learning to summarize the variability of a region of the brain in a two-dimensional map. And there it has been done not using any recognition of the structure, just providing a crop of the brain, a raw crop. This has been done from 20,000 subjects from the UK Biobank cohort. And after that, we project in the same representation of the normal population variability, some preterms so it's preterms from the DHCP, in fact, so with different age of birth. And the interesting thing is that most of the babies concentrate on a specific part of this representation, but the younger they are in terms of birth, the more concentrated they are. Basically, all the red points, they are just there. And we are still trying to understand what it means in terms of what is specific here, even in the adults. But if we use, the modern technique that managed to open the black box, give you some insight about what has been driving the deep learning. The difference, in fact, is in the pole, temporal pole, which makes a lot of sense if you think about preterms. And if you compare these preterms here to standard adult brains, it's like if, in fact, the supratemporal sulcus was not developing normally in the pole. But this is a work in progress. And I will stop there. And I'm really eager to trigger more collaboration with clinical issues, because this is the only way to get some sense out of these tools.